Hello and welcome everybody to my channel Omni Bio and today we will be discussing the part 2 of DBT JRF entrance exam question paper conducted in the year 2017. So coming to the first question. Asynchronous animal cells were stained with fluorescent DNA binding dye analyzed by flow cytometry. The histogram of the cell count versus fluorescence intensity is shown below with or without treatment. Now this is fluorescence intensity graph. These are the cell count. This is for untreated and this is for treated. So first the cells are in the G1 phase, that is G0 G1 phase, that is this one. Then they have entered the S phase, that is over here, this region, and this is the G2 and the M phase. Now we see that the G2 M phase there is increased. G2 or the M phase has increased upon treatment. So the answer here is option C. The treatment increases the proportion of G2 to M phase. Answer is option C. Next question. Which one of the following EIF2 alpha kinases get activated during unfolded protein response in UPR in mammalian cells? So the unfolded protein response in cellular stress is a cellular stress response related to the endoplasmic reticulum stress. The UPR is activated in response to an accumulation of unfolded or misfolded protein in the lumen of endoplasmic reticulum. So PERK or PKR like endoplasmic reticulum kinase. So that is the one which is getting activated during unfolded protein response in mammalian cells. Then next one. E. coli RNA polymerase has 6 subunits, 2 alpha, 1 beta, 1 beta dash, 1 omega and 1 sigma. Which among these subunits imparts specificity of transcription of a gene and how? So RNA polymerase binding in bacteria involves the sigma factor recognizing the core promoter region containing the -35 and -10 elements. So sigma by binding both -10 and -35 sequences. Answer is option B. Then the use of guide RNA to bind DNA and target the double strand break at a specific site is a feature of genome editing by CRISPR-Cas system. Answer is option C. Then epidermolysis bulbosa simplex disease is caused by mutation in which one of the following genes? It is keratin. Answer is option B. Next one. The genes that have been used for reprogramming somatic cells of an adult mammal to stem cells called induced pluripotent stem cells are KLF4, SOX2, OCT4 and CMYC. These code for. So the iPSC technology or induced pluripotent stem cell technology pioneered by Shianka Yamanka, who showed that in 2006 that the introduction of spore-specific genes named MYC, OCT3, SOX2 and KLF4 collectively known as Yamanka factors encoding transcription factors could convert somatic cells into pluripotent stem cells. So these are transcription factors. Answer is option A. Following fertilization, zygote divides rapidly to form a large number of cells within a short time through a process known as cleavage. In some organisms like sea urchin, the cells of the early embryos skip some stages of the cell cycle in order to achieve this. Which of the following steps may be skipped? So it can be G1 and G2 phases that may be skipped. Answer is option B. Then cell cycle regulatory genes were originally discovered by Paul Nurse in yeast using genetic approach involving temperature sensitive mutant screening. Which of the following was used to identify cell cycle regulatory genes CDC? So it was cDNA complementation assay. Answer is option B. Then binding is a protein that is expressed in the tip of sperm head, that is acrosome, and is important for sperm egg interaction. Its distribution pattern changes during sperm maturation. Which one of the following methods may be used to monitor the expression pattern of binding in a simple light microscope? So it is immunocytochemistry. Answer is option C. So immunocytochemistry, it allows researchers to evaluate whether or not cells in a particular sample express the antigen. Here it is binding in the question. In cases where an immunopositive signal is found, immunocytochemistry also allows researchers to determine which subcellular compartment are expressing the antigen. So here it is binding protein. So which subcellular compartment is expressing the binding protein can also be studied using immunocytochemistry using under a simple light microscope. Then next one. Which of the following mechanism converts CERBB EGFR to an oncogene? So it is truncation. Answer is option B. Then mid-blastula transition is a characteristic process that occurs in early embryogenesis of organisms like amphibians whose eggs are large. Mid-blastula transition refers to. So at this stage, the zygote starts producing its own mRNAs that are made from its own DNA and no longer uses the maternal mRNA. The zygote start producing at the mid-blastula transition, the zygote start producing its own mRNA that are made from its own DNA and no longer uses the maternal mRNA. The maternal mRNA are then degraded. Since the cells are now transcribing their own DNA, this stage is where expression of paternal genes is first observed. So they have asked mid-blastula transition. It is a characteristic process that occurs in the early embryogenesis of organism whose eggs are large, and mid-blastula transition, it refers to transition of gene expression from maternal to zygotic. Answer is option B. Then next one. Introns which are self-splicing and do not require any cofactor for their splicing are present in the primary transcript of. So they are present in the primary transcript of all the options A, B, C and D. miRNA, nuclear mRNA, tRNA, mitochondrial mRNA. So answer is A, B, C and D. Then next one. The phenomenon of transfer of traits from a man to his grandson through his daughter, it is known as X-linked transfer or crisscross inheritance. Answer is option B. If the doubling time of a prokaryotic cell becomes progressively shorter, then it can be predicted that the ribosome concentration will. So as the doubling time becomes shorter, the ribosomal fraction is predicted to increase such that the ribosomal fraction times the doubling time is a constant, reflecting the total proteome concentration. So ribosome concentration increases. Answer is option C. Then how many DNA molecules of 6 base pair length are possible where the first base is a purine and the last base is a pyrimidine? So in the first and the last places only purines or pyrimidines can be present. So 2 factorial for those two, first and the last places. Then 4 places in between can be occupied by either A, T, G or C. Hence 2 factorial into 4 C1 into 4 C1 into 4 C1 into 4 C1 into 2 factorial. Okay, so 2 factorial is for first and the last places, only purines or pyrimidines. In between the two factorials, you have to write 4C1, 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 4C1 for ATGC occupancy. Then you will 4 is the total number of distant elements. Here ATGC. 1 is the number of elements drawn or chosen at a time. So 4C1 is equal to 4 and 2 factorial is equal to 2. So if you multiply, you will get 1024. Answer is option D. Then next one. A recombinant protein is expressed in E. coli under T7 promoter at 37 degree Celsius. However, no biological activity is obtained in the cell lysate. If the same experiment is carried out at 25 degrees Celsius, the cell lysate shows a reasonable biological activity. The most probable explanation for this is, so the recombinant protein is expressed in E. coli under a T7 promoter at 37 degrees Celsius, and at that temperature it didn't show any biological activity. But when the experiment was carried out at 25 degrees Celsius, the cell lysate shows biological activity. So answer here is the recombinant protein is properly folded at low temperature. At higher temperature, the protein got denatured. That is why it did not show activity at 37 degree Celsius. Coming to the next question. IPTG is used as an inducer in the T7 expression system for recombinant proteins expression in E. coli. This is because, so the T7 promoter, so this is the E. coli cell, it has got the PET plasmid that is having the T7 promoter, and then you have got the bacterial chromosome which is having lac UV5 promoter for the synthesis of the polymerase gene. No, sorry, for the synthesis of the RNA polymerase protein. Now the T7 promoter, it is acted upon by T7 RNA polymerase to drive the expression of the gene of interest. Then there is a lac operator. This is the lac operator sequence just downstream of the T7 promoter to block the transcription from the T7 promoter, just like your lac operon. And the PET plasmid, it also carries the natural promoter and coding sequence for LAC-I. This is the LAC-I, that is the repressor. Now LAC-I protein, it can act on the lac UV5 promoter in the host cell and repress the expression of the T7 RNA polymerase gene, and also it can act onto this particular operator and block the transcription and translation of the gene of interest, this LAC-I present in the vector. Now if you add IPTG, it blocks the inhibitory action of LAC-I, thereby inducing expression of T7 RNA polymerase and also removing the LAC-I inhibition of the gene of interest. So the answer here is option A, that is availability of special E. coli cells which have T7 RNA polymerase gene integrated into their genome under a regulatable promoter. So IPTG is used as an inducer in T7 expression system for recombinant protein expression in E. coli. This is because availability of special E. coli cells which have the T7 RNA polymerase gene integrated into their genome under a regulatable promoter. Answer is option A. Then pyrosequencing derives its name from the fact it detects pyrophosphate release during base incorporation. Answer is option B. Now, which of the following is a reason that genetics used mitochondrial DNA to study the relatedness of animal population? So, mitochondrial DNA, it is transmitted from mother to the child and it is free of recombination. Answer is option B. Lyophilization is a method used for the preservation of microbes because. So the basic principle of lyophilization is sublimation, in which conversion from solid directly to gas occur. Lyophilization or freeze drying of bacterial culture stabilizes the culture for long-term storage while minimizing the damage that may be caused by strictly drying the sample. So the answer here is moisture is removed by sublimation. Answer is option A. Stoichiometric matrices in metabolic pathways are used in flux balance analysis. Okay. Now this is a stoichiometric matrix. Now this one. This one is the stoichiometric matrix for this particular pathway, that is fructose 6-phosphate to fructose 1,6-biphosphate of glycolysis pathway. Part of a glycolysis pathway is represented here, and this is the stoichiometric matrix of the pathway. So it is a mathematical representation of the large number of reactions obtained for a specific organism. Then in stoichiometric pathway, there is a representation of rows and columns. So a cell with non-zero value, that is either 1 or -1, the others are 0. So a cell with a non-zero value in the matrix represent the stoichiometric coefficient of the corresponding metabolite in the corresponding reaction. So R2, it is 1, so it is F6P, that is fructose 6-phosphate. Now, a positive value means that the product, that it is a product, whereas a negative value indicates a substrate. So R1, if you see, that is row 1, if you see, fructose 6-phosphate is -1, so it is substrate, and R2 is fructose 6-phosphate is a product also. So it is acting as a substrate as well as a product, so it is showing a reversible reaction. So that is what this matrix represent. What are the products obtained, what are the substrates, whether the reaction is reversible or irreversible can also be known from this particular matrix. Several quantitative analysis methods have been developed based on stoichiometric matrix of the metabolic network, such as flux balance analysis, elementary flux mode analysis, and extreme pathway analysis. Then next one. The main difference between domain and motif in protein structure is, so the domain can remain stable independent of the rest of the protein while motif cannot. Answer is option A. Select the best algorithm to do pairwise alignment when two proteins are very different in length. So it is Smith and Waterman. That is for local alignment. Then, from literature, it is known that the length of an E. coli bacterium is 1 micrometer with a standard deviation of 0.1 micrometer. After treatment with chemical X, the mean length of 100 cells is 1.1 micrometer with standard deviation of 0.1 micrometer. What will you conclude from this experiment? So, treatment with chemical X has increased the length with a statistical confidence of more than 99%. Answer is option A. Then which one of the following techniques cannot be used to remove salt from protein solution? So dialysis, it is commonly used laboratory scale process to remove salt or reduce the salt concentration from a solution. Ultrafiltration is used for removal of salt. Desalting and buffer exchange use gel filtration. Chromatography to separate soluble macromolecules from smaller molecules. So which one is not used to remove salt from protein solution? It is ion exchange chromatography. Answer is B. The next one. The jelly roll in protein structure is, jelly roll or Swiss roll fold is a protein fold or super secondary structure composed of 8 beta strands arranged in two TWO2 4-stranded sheets. So the answer is formed from 8 beta strands. Answer is option D. Which one of the following is not a structure or structurally derived database? So the answer here is prosite. It is not a structure or structurally derived database. Prosite is a protein database and it consists of entries describing protein families, domains and functional sites as well as amino acid patterns and profiles in them. So the answer here is option B, that is prosite. Then which of the following is true? In the threading approach for prediction of protein structure, the optimization is used for. So threading, it is a method of tertiary structure prediction method in protein. So what you do, first you take the protein sequence, then you search the library of templates or known structures. They are the PDB structures. Then you search over there for best composition of sequence as measured by energy function. Once all the candidate structure together with their scores are gathered, the lowest energy one is accepted as the structure prediction of the protein. That is how you do threading. So sequence to sequence alignment, no. Generation of profile by converting three-dimensional structure to one-dimensional string, no. Alignment of sequence to structure which minimizes energy of the target sequence in the template fold. That is correct. Answer is option C. Then next one. The statistical significance of the blast hit is obtained using extreme value distribution. Answer is option A. The next one, what is referred by the term K in the following energy expression? E is equal to half K B minus B0 square, where B and B0 refer to the bond length and reference bond length respectively. So it is K is stretching constant for bond length variation. Answer is option. The estimates of a number of false positives from a blast search can be made using percent identity e value. So e value, it gives an idea about false positives. Short identical sequences may have high e value and may be regarded as false positive hits. This is often seen if one searches for short primer regions, small domain regions, etc. Can be made using e value. Answer is option C. The length of beta hairpin motif is usually. So the beta hairpin, it is a simple protein structural motif, and the motif consists of two strands that are adjacent in primary structure, oriented in anti-parallel direction and linked by a short loop of 2 to 7 amino acid residues. So these are the anti-parallel beta strands and they are linked by a loop over here which is 2 to 7 amino acid residues. So answer is option A. The next one, a scoring function is used in which one of the following drug design technologies? So scoring function, it is an additive function that includes representation of various interaction between a ligand and a target. These representations describe the electrostatic, hydrophobic, solvation and hydrogen bonding interaction between the ligand and target. So scoring functions are used by docking programs for estimating the binding affinity between the ligand and target as well as for predicting the native structure of protein ligand complex. So scoring function is used in which one of the following drug design technologies? It is molecular docking. Answer is option B. Blossom matrices are based on highly conserved regions in a series of alignments forbidden to contain gaps. Answer is option B. The next one. ProDOM is a comprehensive set of protein domain families automatically generated from. Answer is Uniport Knowledge Database. Answer is option B. Which one of the following methods used to find evolutionary trees is also referred to as minimum evolution method? So minimum evolution, it is a distance method employed in phylogenetic modeling. It shares with maximum parsimony that the tree with the smallest sum of branch length estimate is most likely to be a true one. So which one of the following methods used to find evolutionary trees is also referred to as the minimum evolution method? It is maximum parsimony method. Answer is option B. Then Z-score equals score of alignment minus mean divided by standard deviation. In the sequence logo of the type given below, the sizes of the letters are proportional to. So these sizes, G is bigger here, T is small, C is small, A is again taller than G. So you get something like this graph. So what does these sizes of the letter represent? So they represent frequencies of the respective residues in the sequences. Answer is option C. Then next one. Of a population of cells undergoing meiosis, 1% of the cells undergo recombination between genes A and B. What is the distance between the two genes? It is 0.5. Answer is option C. Then which one of the following combination of marker, genes and promoters cannot be used for selection of transgenic plants under in vitro condition? So you cannot use non-conditional negative selection marker genes under CAMB35 as promoter. Answer is option D. Then next one. Several experiments have shown that leaky deregulated expression of reporter gene, that is GUS, from plant promoters in agrobacterium cells. Which one of the following approaches would be most useful in restricting transgene expression in transformed plant cells and prevent their expression in agrobacterium? This is a repeated question. So a plant intron was introduced into the GUS gene. In transgenic plants containing this chimeric gene, the intron is spliced efficiently, giving rise to GUS enzymatic activity, whereas no GUS activity was detected in agrobacteria containing this construct due to lack of a eukaryotic splicing apparatus in the prokaryotes. So answer is use of introns within the transgene sequence. Answer is option D. Then next one, a T0 transgenic plant showing 2 copies of tDNA on southern analysis segregated in a 3:1 ratio for the transgenic is to non-transgenic phenotype among T1 progeny obtained by self-pollination. Which one of the following statements best explains this observation? So, it is a single gene. If it is a single gene integration, then the ratio of T1 is 3:1. But in the question, 2 copies of tDNA segregated in 3:1 ratio, showing that they are linked. So the answer here is option B. The plant contains 2 linked copies of the transgene. Answer is option B. Then which one of the following statement is correct? All the virulence gene of agrobacterium tumefaciens are expressed in a constitutive manner. No, or not all the virulence genes, only 1 or 2 are expressed in a constitutive manner, the other it is expressed in inducible manner. Then opines are source of iron for agrobacterium cell. Opines are source of carbohydrate derivative that serve as nutrient source for the agrobacteria. Then one agrobacterium cell can generate only one tDNA molecule for transfer into the host cell. That is wrong. Then integration of tDNA in the genome can mutate or modulate endogenous plant genes. That is correct. That was seen in the case of petunia. They tried to express more color by overexpressing a chalcone synthase gene, but it resulted in endogenous gene silencing, that you got patches. White patches against a purple background. That is what the result they have obtained. So answer here is option D, that is integration of tDNA in the genome can mutate or modulate endogenous plant gene. That is correct. Now next one. Which one of the following statements related to transgene silencing in plants is incorrect? Now transgene silencing always occurs in T0 individuals and never in subsequent generations. That is the incorrect statement. It occurs in subsequent generation also. So coming to the next question. 2 independent transgenic plants, one with single copy of gene A and another with single copy of gene B, were expressed under the same seed-specific promoter. In transgenic plants, seed formation on self-pollination was similar to that of untransformed plants. So when homozygous plants with gene A, that is male parent, were crossed with homozygous plant with gene B, that is female parent, viable seed formation did not occur, although pollen production was normal. What could be the possible reason for this observation? So option A. Product of gene A is lethal to the male gametophyte. If that was so, then pollen production would not have been normal. It is given in the question that pollen production was normal. So A is not correct. Interaction between products sorry, interaction between products of genes A and B is lethal to the zygote. That can be the reason, because they both, that is gene A and gene B, they are seeds, they are under the control of seed specific promoter. So in the seed, the viable seed formation did not occur although pollen production was normal. So even when pollen production was normal, viable seed did not, production formation did not occur. So interaction between products of genes A and B is lethal to the zygote. Interaction between products of genes A and B is lethal to the zygote. So in the seed, the genes A and B got expressed and their interaction resulted in a formation of a product that was lethal to the zygote. That is why you did not get viable seeds. Then interaction between products of A and B is lethal to the male gametophyte. That is not correct. Product of gene B is lethal to the zygote is also not correct. Answer is option B. Then next one, the range of transformation frequencies obtained is in independent transformation experiment using 4 different constructs with a different combination of promoters and selection marker genes is given below. In the absence of any other factors, which of the following statement is not acceptable as a logical conclusion based on the above data? So first one, use of strong promoters for expression of selection marker genes can increase transformation frequencies. So when BAR gene and NPT2 gene is placed under CAMB35S promoter, that is a constitutive and a strong promoter, the transformation frequency is higher than that of BAR gene and NPT2 gene under the control of NOS promoter. You can see from the values over here, transformation frequency is more for CAMV35S promoter than for the NOS promoter. So use of strong promoters for expression of selection marker genes can increase transformation frequencies. That is correct. Then variation in expression levels between NPT2 and BAR gene is not influenced by the NOS poly A signal. Yes, of course, it is not influenced by NOS poly A signal. In all 4 cases, NOS poly A signal is there. It is not influenced. Production of herbicide-resistant plants at high frequency can be achieved with lower expression levels of the transgene. That is the answer. Then, use of kanamycin as a selection marker appears to be more favorable for production of large number of transgenic plants. Kanamycin marker is NPT2. So, use of kanamycin as selection marker appears more favorable for the production of large number of transgenic plants. See, if you see over here, instead of the BAR gene for selection of transformants, NPT2 gene is giving more number of transformants, whether it is CAMV35S or NOS promoter. So option D is also correct. So what is not correct is option C. Answer is option C. Then next one. Callus mediated regeneration is not preferred for micropropagation because the direct regeneration protocol is ideal for micropropagation to obtain large number of elite seedlings because the buds and the shoot originate directly from the epidermal cells bypassing the callus phase. And if callusing is there, it can lead to generation of variants. So answer is option C, that is it leads to generation of variants. Then next one, in a transgenic plant, the phenomenon of co-suppression is due to when plant carries a transgenic copy of an endogenous gene, both the genes get silenced. That is co-suppression. Now example is, I have told you one already. Sense transgene meant to overexpress the host chalcone synthase A gene caused the degradation of the homologous transcripts, and the loss of flower pigmentation. That is when to increase the flower color of petunia, chalcone synthase gene was overexpressed. But what happened was this overexpression, it resulted in the silencing of the endogenous gene also. So co-suppressed petals, they showed up as white in a purple background. So the answer here is similarity between the transgene and the endogenous gene sequences. In transgenic plant, the phenomenon of co-suppression is due to similarity between the transgene and endogenous gene sequences. Then next one. In inbred lines, gene and genotypic frequencies are maintained by growing them in random mating without selection. Answer is option D. Homozygous plants from biparental mating for development of mapping population can be obtained in a short time by development of double haploids. Answer is option B. Double haploids will give rise to homozygous plants. Which one of the following is not required for QTL analysis in association mapping? Genetic linkage map is not required. Answer is option C. Which one of the following hormones promote production of seedless grapes? So it is GA3. Answer is option D. Then next one. Which one of the following molecular markers is associated with bacterial blight resistance in rice? It is XA21. Answer is option C. Match the proteins in group 1 with members in group 2. Alkaline phosphatase. It is periplasm. F0 component of the ATP synthase. It is inner membrane. Alcohol dehydrogenase. Cytoplasm. Pectinase. Extracellular. So the answer here is option A, that is 1S, 2R, 3Q, 4P. Then application of herbicide phosphonothricine results in the death of the plants due to accumulation of. So herbicide phosphonothricine inhibits glutamine synthetase, a key enzyme in nitrogen assimilation, and thus causing ammonia accumulation, glutamine depletion, and eventually plant death. So there is accumulation of ammonia. Answer is option B. The next one. Which one of the following is not a feature of Agrobacterium tumefaciens E protein? Its synthesis is induced by Vir-G protein. Yes. It contains nuclear localization signals. Yes. It protects tDNA from being destroyed by plant defense mechanism. Yes. It is component of T pilus. Of the Vir genes, the 11 genes of the B operon are involved in the synthesis and assembly of the T pilus, with the product of Vir B2 gene being the subunit precursor of the T pilus. So it is not Vir E protein, it is Vir B. So answer is option D. The next one. Transgenic potato plants with high amylostarch were developed by suppression of starch branching enzyme. Answer is option A. So next question. Developing cisgenic disease resistant apple is advantageous over conventional breeding because cisgenic is the genetic modification of a recipient plant with a natural gene from a crossable sexually compatible plant. Cisgenic plants are presumably considered safer than those produced through conventional braided plants because of the lack of linkage drag. So the answer here is option B. There is no linkage drag. The next question. For the development of selection marker-free transgenic plants by co-transformation using Agrobacterium, the marker genes are. So the gene of interest and selectable marker gene from different tDNAs were able to be simultaneously integrated in T0, and marker-free plants can be yielded after their subsequent recombination and segregation in T1. So it is segregated out in T1 generation. Answer is option C. Breakdown of Bt mediated insect resistance in crops can be delayed by. So this is a repeated question. I have given the explanation in the previous question paper, I think 2016, I think explanation there. The answer is option A, that is refugia strategy. You can refer that for explanation. Then next one, which one of the following is matched incorrectly? So bar gene resistance to virus. Bar gene confers resistance to herbicide by Bialafos. So answer is option C. Which one of the following would produce androgenic haploids in anther culture? So young microspores, they are haploid, all other options are diploid. Answer is option D. Male sterility in plant is induced by expression of TA29 Barnase PA cassette in tapetum. Answer is option C. To achieve complete separation of two pharmaceutical compounds shown as two peaks in the chromatogram, the relative separation RS would be. So, the goal for any chromatographic operation is the separation of two or more species. The measure of the ability of a column to separate species is known as peak resolution or relative separation RS. It is defined by the equation that they have given over here. 2Vr2 minus Vr1 by Wb1 plus Wb2. Vr1 and R2, they are the retention volume for different molecules. This is it, retention volume when they separate out. So Vr2 is greater than Vr1. And Wb1 and Wb2 are the width of each chromatographic peaks. This is Wb1 and Wb2. To completely separate two molecules, RS must be equal to or at least 1.5. So RS greater than 1.5. So answer is option B. Then the anti-cancer drug Halichondrin D is isolated from. It is isolated from corals. Then heparin is sulfated polysaccharide. The only naturally transformable marine cyanobacteria is Echinellum species. Answer is option A. Marine chemosynthesis is mainly based on carbon dioxide. Answer is option B. Which one of the DNA replication blocking agent produced by sponges? Spongiosides. Eutrophication in aquatic ecosystem is due to reduction of oxygen. Eutrophication is the process by which an entire body of water or parts of it become progressively enriched with minerals and nutrients, particularly nitrogen and phosphorus. Eutrophication leads to increase in the amount of plant and algal growth and in the water, and this will result in lesser availability of or reduction of oxygen levels. Then halotolerant and halophilic microbes can be isolated from dash and dash respectively. So they can be isolated from option A, that is estuary, and C. Heavy metal pollutants like cadmium and mercury inactivate enzymes by interacting with cysteine. Answer is option A. Which of the following class of enzyme initiates aerobic degradation of aromatic pollutants like naphthalene in bacteria? Degradation, that is oxidoreductases. Answer is option A. Which of the following organism is not able to perform light harvesting reaction? So answer is option A, azospirillum. In the soil environment, which one of the following factors is responsible for the biotic stress on the microbial community? So, biotic stress is a stress that occurs as a result of damage done to an organism by other living organisms such as bacteria, viruses, fungi, parasites, etc. So, the answer here is option D, that is microflora. A to C are abiotic. Then next one, match the enzymes in group 1 with their appropriate role in the environment from group 2. Lacase is lignin degradation. Catalase is neutralization of toxic oxygen species. Nitrogenase is nitrogen fixation. Cytochrome P450 is xenobiotic detoxification. So the answer is option A, 1R, 2S, 3Q, 4P. Which of the following types of mutation would usually not be detected in a molecular diagnostic test that is based on sequencing each exon of a gene individually from a male suspected of having X-linked disorder? Answer is inversion of a part of the gene. Answer is option D. A couple came for counseling following 3-trimester miscarriages and underwent chromosomal analysis. The man was found to be have pericentric inversion with a karyotype 46XY INV8P12Q22. Which one of the following would be the correct conclusion? This rearrangement might lead to chromosomal imbalance in an offspring and could explain multiple miscarriages. Answer is option C. And option D, the rearrangement might cause dicentric or acentric chromosome in an offspring which would probably not be compatible with survival, that is seen in the case of paracentric inversion. The next one, unequal crossing over between two repeats can lead to an LDL receptor gene with an internal deletion or duplication. Based on this information, which of the following must be true? So it is the LDL receptor gene contains at least 2 allele repeat sequences. Answer is option D. Then next one, linkage analysis is performed in a large family with an autosomal dominant hemolytic anemia using a polymorphic marker with the beta globin locus. The load score at Q is equal to 0 is negative infinity. Load score at Q is equal to 0.01 is minus 4.5. You conclude that the disorder in this family is. So load score is an acronym for log of the odds. Load score actually refers to a numerical result when estimating whether two genes or a gene and a disease are linked to one another. So load score are most often used to describe the data one gets out of family studies when you are looking at large families and an inheritance of traits or diseases within the family. So positive load score favor the presence of linkage, whereas negative load score indicate the linkage is less likely. Now negative load score indicate that the linkage is excluded. Thus it is very unlikely that the gene for hemolytic anemia is linked to a region within or close to the beta globin locus. So answer is not due to beta globin gene mutation. The disorder in this family is not due to beta globin gene mutation. The next one, in Li-Fraony syndrome, most frequent mutation occurs in. So this condition is most commonly caused by mutation in the gene called TP53, which is a genetic blueprint for the protein called P53. So the answer here is option A, that is P53. Then which one of the following promoters is most efficient for transgene expression in mammary gland of livestock? Beta-casein. Answer is option A. Epinephrine is used along with local anesthetic because of its alpha adrenergic receptor agonist properties. Answer is option A. Founder effect and bottlenecks are forms of genetic drift. Answer is option D. The next one. In commercial farms, embryos are routinely recovered by non-surgical methods. Which day of the embryo recovered on which day are the embryos recovered from the donor cow after the onset of estrus? So 7. Answer is option B. Human mesenchymal stem cells. Human mesenchymal cells are non-hematopoietic multipotent stem cells with the capacity to differentiate into mesodermal lineage such as osteocytes, adipocytes and chondrocytes, as well as ectodermal and endodermal lineages. So can differentiate into few types of cells. Answer is option B. Vectors are important in transmission of diseases. Match group 1 with group 2. Louse, epidemic typhus, ticks, Lyme disease, mite, scrub typhus, mosquito, West Nile fever. So 1D, 2C, 3B and 4A. Answer is option A. Which one of the following is an inhibitory neurotransmitter? GABA. Huntington's disease is clinically characterized by chorea, abnormal involuntary movements. Which part of the brain is responsible for this phenotype? It is the basal ganglia. Next one. Alpha-amanitin is a fungal toxin which inhibits eukaryotic RNA polymerases. The 3 eukaryotic RNA polymerases show differential sensitivity to this toxin. Which one of the following order is the correct with respect to the sensitivity towards alpha-amanitin? So it is the RNA polymerase 2 greater than 3 greater than 1. Sorry. The next one. A primary cell culture can be transformed into a cell line by all except hepatitis B virus. Then next one. Assessment of the extent of DNA double-strand break in cultured human cells following exposure to ionizing radiation can be done by quantitation of an immediate result of ionizing radiant exposure in the DNA double-stranded break that can be detected with an antibody to gamma H2AX, a phosphorylated form of histone H2A variant. H2AX, which forms rapidly at the site of DNA double-stranded breaks. And H2AX is required for the concentration and stabilization of DNA repair protein and play a role in both non-homologous end joining and homologous recombination repair pathways. So phosphorylated histone H2AX quantification. Answer is option D. Then next one. There are reports of more than 100 mutations at different sites of factor 9 that manifest hemophilia B. This is an example of allelic heterogeneity. Answer is option B. In a randomly breeding population, an autosomal recessive condition affects one newborn in 10,000. The expected frequency of carriers will be. So according to Hardy-Weinberg law, P square plus Q square plus 2PQ is equal to 1. P square and Q square frequencies of homozygous dominant and homozygous recessive individual respectively 2 PQ represent frequency of carrier, P and Q represent frequency of dominant and recessive allele respectively. So Q square is equal to 0.0001 or 1 by 10,000 and Q is equal to 0.01. P plus Q is equal to 1. P is equal to 0.99 and they have asked for expected frequencies of carriers. So 2 into 0.01 into 0.99, you will get somewhere around 2%. So accordingly, 1 person in 50 will be the carrier. So 1 in 50. Answer is option B. Specificity factor which activates ubiquitin ligase activity of APCC during suppression of sister chromatids is. So the anaphase promoting complex cyclosome APCC is a multi-subunit ubiquitin protein ligase that targets for degradation cell cycle regulatory proteins during exit from mitosis and in the G1 phase of the cell cycle. During metaphase to anaphase transition, APC activity requires association with activator subunit CDC20 that contribute to substrate binding. So specificity factor that activates ubiquitin ligase activity of APCC during suppression of sister chromatids is CDC20. Answer is option B. Then next one. In diploid organism, loss of function mutation produce dominant phenotypes when there is. So when there is haploinsufficiency. Haploinsufficiency, the situation that occurs when one copy of a gene is inactivated or deleted and the remaining functional copy of the gene is not adequate to produce the needed gene product to preserve normal function. So haploinsufficiency may arise from a de novo or inherited loss of function mutation in the variant allele such that it produces little or no gene product. Answer is option D, that is haploinsufficiency. In Mendel's dihybrid cross experiment, if the two selected traits were on the same chromosome and assuming that there is no recombination, what would be the expected genotypic ratio in the F2 generation? So YYRR is the dihybrid cross experiment and they have asked what would be the expected genotypic ratio in the F2 generation. If the two selected traits were on the same chromosome linked and assuming that there is no recombination. So you will get 4 gametes and upon crossing you will get the ratio 1:2:1. Huntington disease is caused by. So Huntington gene provides the genetic information for Huntington protein. Expansion of CAG repeats in the gene coding for the Huntington protein result in an abnormal mutant protein which gradually damages the brain cell. So expanded trinucleotide repeat in the coding sequence. Answer is option C. Then next one. Microscopic evaluation of a post mortem brain exhibits hyperchromatic areas when stained with glial fibrillary acidic protein. This suggests activation of. So glial fibrillary acidic protein is an intermediate filament 3 protein uniquely found in astrocytes in the CNS, non-myelinating Schwann cells in the PNS and enteric glial cells. So answer here is option A, that is astrocytes. Coming to the next question. The following pedigree shows the inheritance of a very rare human disease. What is the most likely mode of inheritance for the disease trait and what is the probability that the second child of 3-2 and 3-3 will be a son and will also have the disease? So the answer here is it is autosomal dominant. Now why it is autosomal dominant? Because every affected person has got an affected parent. See here this person is these persons are affected and they have got an affected parent. Here also this parent is affected, the child is also affected. Then each child of an affected parent has 50% risk to inherit the disease. So here one child is normal, the other child is affected. Here also one child is normal, the other child is affected. Here also one child is normal, the other child is affected. To inherit, then unaffected family members do not transmit phenotype to children. So these parents, they do not transmit the phenotype to their children. That if you see over here, you can make out that the gene doesn't come from unaffected parent. Then male and females are equally likely to transmit the disease. There is no X-linked dominant inheritance or recessive inheritance here because there is no male to male transmission. So probability that the second child is a male is half. Probability that he will have the disease is half. In the case of 3-2 and 3-3, that is this one. A and small. I have rewritten it over here. So capital A, small a, small a. You get capital A, small a, small a, small a. So probability that the child is male with the disease is born is 1/4 or 0.25. So the answer here is option D. Then next one. Parasite number parasite numbers in the blood of a patient with trypanosoma brucei, that is sleeping sickness, shows the following pattern. Parasites isolated from population 1 and 2 were found to be antigenetically non-cross reactive. The parasite distribution seen may be due to. So it may be due to antigenic switching. Answer is option D. And antigenic switching is the alteration of a microorganism surface antigen through genetic rearrangement to elude detection by the host immune system. So here, as the number remains the same as the days pass by, because the number of parasites in the blood of the patient remains the same. There is no decrease in the number of parasites in the blood of the patient as the days pass by, showing it is antigenic switching. Answer is option D. So thank you for watching my channel. Please do like, share and subscribe. Thank you.